Before I was talking, this is what I was trying to say before is something that is generic for all the NGS kind of pipelines. Now I'm moving something that is specific to the RNA-seq. Okay? And I think, first thing, the pipeline is something like that, the one I'm going to show you. I mean, BCL to FASTA-Q we talked before, FASTA-Q to SAM, this is the mapping. I will talk a bit. Then we need to quantify, then we do differential analysis, then we do the functional analysis. Question is, do you find anything missing here? Filtering, exactly. That was the point I was making to make. I mean, we make a lot of things here before, like, okay, how good is that? Then I was talking a lot about quality controls. In reality, we take quality controls every step we do. And this is really, really important. The ones that we did here, I think it's more or less clear, quality of the reads, etc. And here we will also show some of the quality controls. That's important, but at any stage we should have. In this one, I mean, when you identify the genes or when you do the differential expression, quality control is more like, okay, what is the distribution of my p-values, or even more, what is my top genes? Are the ones that I'm expecting? I have any positive control that I'm expecting to happen? This is, I mean, we need to check on them. And maybe following your comment, that's kind of all the quality controls are related. So if you are being like less conservative here, which is not good, but then you come here and say, yes, but I get exactly what I was expecting, then okay, I should take it carefully, but still there is some information I can make use. I will put all the quality controls in the perspective. So let's go step by step. So mapping every sequence. The problem of mapping, everyone feel confident when I say mapping to the genome. So basically, we have this situation when we have the read, and I need to find where it may end here, right? Originally, you have the sequence, and you have in the BLAST algorithm to try to identify where it was. This is one situation when we have a single end read, and then we have a parent read. So we have like one extreme and the other extreme, and something in the middle. And look here. Like this is an example of what we are looking into, right? That's kind of the very intuitive, very trivial thing. Algorithms for mapping, there are huge of them. And it's scary because it's still kind of coming out, some of them saying, well, we are better and we should do that. And it's still kind of room for improvement. This is what it's quite clear. I think if we take, it's just taking small talk about what Bowtie and BW are doing. Originally, we have one problem that was you have one sequence, like the BLAST, and you kind of want to find something in the entire genome. And then you are like checking locations, or you have this Smith-Waterman algorithm. Here is a really complex, right? Because we have thousands and thousands and thousands of them. And sometimes we don't want to be, we want to be extremely accurate, sometimes not, sometimes we want to be a bit more heuristic, and we want to try to find where they end up in the genome. So just as an intuition of how it works, they create an index of the genome. It actually creates a tree of the genome. And it's actually similarly to the methodologies that the word processing is doing when you are searching for a word and it kind of finds it really fast. It's basically similar, it's not the same. But if you start with A, you look for what is, this will be A, then C, then C, then T, then T, then blah, 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 blah. Okay. You create a tree saying AC, CC, or AB, AT, AC, AG, and then you are set of those sequences you are putting in this tree where they are ending. So you reorganize your genome as a tree. What happens? That when you have this read, it's kind of easier to look in the tree. Because I look into this where C, where A is, where this one, I follow, and at the end I say, okay, and this one, where is in the genome? Where can I find it in the genome? So it gives you like a really, really fast way of searching for them. It's because, I mean, I don't think we are really interested, or we don't have really time to go into that more, but I put some reference here for the ones that are interested in reading that. Sometimes the important thing to comment here is you can decide on the quality. Those methods are a bit heuristic of how they are searching. You can say, I want to spend more time and really, really go for high quality, or I want to find, if I find one that is good enough, I'm done. Or I want to find all the places in the genome with this read can map. What I usually do when I'm doing the mapping is try to be high quality, and if it maps to more than one place in the genome, I discard the read, because you don't want to have same fragment to be mapped to 2 locations in the genome. That's the problem. You will not be able to say, okay, this is gene A or gene B, so we take them out. There were some methodologies before that they were telling if it maps in 2 locations in the genome, you split like half here, and you give half reads. But that was the time when the sequences was very expensive, when you want to make the best of every one of the reads. And now we are in a situation that maybe we can take out some of the things that we are not really sure about. So duplications in the mapping locations, it's kind of, they can map to more than one place, I will take them out. When doing the mapping, this is the ideal situation, right? But this is not perfect. We can have insertions, because every kind of us have a different genome. We can have deletions, we can have mismatches, either because we have a genotype, either because the quality of the read is not that good. So in addition to these 3, you have a quality saying like how good is this mapping. And the good is, if I need to make more insertions and more insertions, maybe it's less likely that something is going to be there, right? If I'm making more deletions, more mismatches, the more I have to change, the worse is going to be the quality. And usually you try to find the best quality weighting every one of those options. I just really recommend reading those papers. So what happens is, after we have this sequence, we go with bow tie or bow tie 2. Now it's a new, kind of a few years ago came. BWA, Novaline in the kind of, for some time, CLC Bio is also mapping. And then we have a new format, because then we need to store more information. And this format is called the SAM files. And this is an example. I want to just show you, because sometimes it's good that you are able to read a bit into the file. So imagine we have a sequence like those ones that I'm showing here. And imagine we have a reference like this one, right? And this reference has only 1 chromosome. So that's easy. Then I need to find a way to say, well, this sequence is mapping in this location. The SAM format will need to store, okay, what is the reference, like in this case, some extra information, and then for the first read, this is my first read, where did it map? In the reference, in the location 7, the quality, some extra information, and then the sequence, and also the quality, and a lot of more things. I don't want you to right now, we are not going to disclose every one of them. I'm just going to show you the ones that, for me, I think are more important. So this is an example of another read. This is more like similar to what the STATegra has. And this is just mapping to the chromosome 20, version of the mapping, some information about it. You can look into this link. You will get all the information if you want to dig more. This is the read I get. This is the chromosome. This is the location. this is the sequence, and this is the quality of the sequence. You can see that the quality of the sequence goes back to what I was saying before. We have the thread 33, thread 64, etc. So the SAM files will tell you also about which one was used for the sequencing. You have information on format version, sequence name, platform. Those are probably two of the ones that are most important. This flag is information about if it is a parent, if it is a single end, if I'm a parent, if my mate has been mapped or not, if I'm a chimera or not, if I have different information. And this is the quality of the mapping. And the quality has different values. It depends on the mapping. Bow tie has different kind of ways of approaching it, and you need to look into each one of them. As brief description is here, this is what I was showing about the flag. Will tell you about the information, like, I mean, if it is multiple segments, if it is properly mapped or not, if it is the other one has been mapped or not, if it's a secondary alignment, so it can map to more than one location, etc. What I want to say with that is this is important after the mapping to filter the things that you want to use. What are the things that we are using in STATegra? Both ends are being mapped. They don't map to different genomic locations with the same quality. So the top quality has only one genomic location, and the quality is kind of strong enough. That's kind of the usual way of approaching it. And like I was saying, important things, this is the chromosome, the position, and the quality. And you can see that here it says something, but it depends on the mapper. We'll give you like different qualities for that. I want you to also think about important aspect. When you are mapping, you can map to the ensemble reference, to the NIH reference, to the different things. Usually, I mean, well, now right now they are kind of in agreement. The problem is the IDs they are using for the chromosomes can be different. Some of them is CHR1 or just 1. And this is trivial, sometimes gives you some problems. So be aware of that. So what is the problem when we are now imagine we have this sequence and this mapping into the entire genome? That's fine, we have this sequence. The problem is when we want to map like to a transcriptome. And just let me show you this pipeline and think about that. We have a gene that has like different exons, right? And then we have our fragments. If we map them and they map inside the exon, it's not a problem because they are mapping to the genome, right? But what happens if I have these two exons and my read is mapping right in the junction? What I see is not the intron, what I see is this part because it has been processed, right? So I can see only this part. This will not map to the genome. I need to split it in two and then map each one to each one of the exons. That's what happens here. This is just a unique read that can maps to two genomic locations, right? This is like this one. The problem is, if I take the entire genome, by default, this will not map directly, because I have the read and I have to split it in two. The question is, how I know where I can split? And those are the splice junctions. We need to create a reference of all those possible splice junctions. Create them and then map to them. How they do that in the top hat? You have different options. One option is I have a reference, so I know the genes, I know the transcripts, I give them, and then they map to the reference, right? The other one is, well, I'm going to use the reads that map not to a splice junction, and they map here perfectly. So this is an exon, this is an exon, this is an exon, and this is an exon. Once I have identified those exons, I can think about the possible junctions. Once I have identified the possible junctions, I create a library of the possible junctions, and I map to the library. And this is how kind of Top Hat is working. You have more information in those papers. So this is how Top Hat is working. It maps to the genome, identifies the exons, then creates the database, and then it maps to it. And the creators of Top Hat are the same ones as the creators of Bowtie. So to map, they use Bowtie. But to create the junctions, to set the database of junctions, this is what Top Hat is for. There are many comments or many papers about Top Hat or many other methodologies. In general, I mean, with my last reading is that maybe there are better algorithms than Top Hat, but when you have a reference genome that is kind of quite robust, Top Hat is making a good job, very good job. What are those? I mean, human, mouse, rat. If you go to rare genomes, maybe you should start thinking about Top Hat is not the best option. But for well-known genomes, Top Hat is good, because it will actually create the database of junctions based on your reference and your data, if you want. So that's how it works. So how we check? I mean, after we do this, we check about percentage of the fragments, so 2 reads that map to the genome, percentage where only 1 read was mapping, duplicate mapping, so the same fragment maps to 2 locations, and the chimeric ones. You know what is a chimeric? Here, it may map to 1 chromosome and to another chromosome. And if we think about, for instance, high-C data or chromatin conformation data, this is the ones that could be quite interesting because it's kind of interchromosomal relations. But in RNA, this is something that we don't want, and we need to take them out. So it's also informing in the sum format about if you have a chimeric or not. Tools for doing that, Picard tools or SAM tools are really kind of the most powerful ones among them. Are you with me here? I think, I mean, more or less, this is the mapping. What I'm going to show now is becoming closer to what will affect our kind of more our analysis. I mean, we are coming into the pipeline of differential expression, etc. Because right now we have reads, right? Question is, we need to assign reads to genes. Because I was telling before, we can map to the genome with no problems. Okay. And then I have this read that is mapping here. This is my read. And I have a gene here. Should I assign this read to this gene? Okay, I will say yes. That's kind of trivial. What happens if my gene is defined from here to here and my read goes also a bit out? Should I consider it to be part of the gene? Sorry? Is that? Maybe it's uniquely mapped. Still, it's uniquely mapped. Will you assign this to the this one? So you. It can be, or maybe this is our reference and this is not really the true. There is an isoform which longer kind of. That's possible. What you will do in this situation? So the read is mapping to the both exons, but it's also mapping to the intron. I mean, in this case, I will be very happy. It's like, wow, perfect. But in this case, I will start in like, but maybe you have some parts that is not, I mean, it can be different reasons that we don't have the right reference. We think this is the gene, but maybe there's another gene that have an exon there. Maybe you have contamination with still not processed kind of mRNA, and then you have still this part. That would be the options, but it's not uncommon that you find introns with levels of expression in the RNA-seq data. Like, I mean, yeah, that's what I'm saying, but that they appear. Sometimes, I mean, I'm not telling that it's like, but some, that's what I'm saying, the possible options. Maybe the reference is not right, maybe we are having like a kind of, we have like with the mRNAs still at this stage and not kind of being spliced. Anyone has other options? Yeah, that's the other. That's what I say. And this is, I mean, we think this is our reference, but maybe it's not. Maybe this is not an intron. For some of the transcripts, it's not an intron. So, I mean, I think, I mean, the problem is you see it, and then you start like wondering, because in many genes you see like perfect, oh, that's the exons, and that's intron is almost nothing. But sometimes you see a read in the introns, or some reads in the introns, or what is that? Is contamination? Is not? I mean, if the intron level is at the same level of the exon, that's a different kind of level of situation where you should be digging into. I mean, why that happened? If it's like the exon is a lot of reads and some of them are here, maybe, I mean, contamination, maybe, or maybe this is not mapping here. This is mapping here, but in reality, because it maps here perfectly, but it can be elsewhere. So I propose more challenge. What do you do here? You have the read here, you have a gene here, and a gene here. Will you assign this to the read to the gene A? I will say yes. But what will you do here? And what will you do here in this case? I think one option is to say, well, I'm going to take the union, so I'm going not to be that conservative, and if kind of overlaps gene A, if overlaps perfectly gene A, if overlaps partially gene A, if it goes to the gene A, here is when I'm not sure, but here it's perfectly to gene A, not to gene B. Okay, gene A. Sorry, this is ambiguous in both of them. Sorry, I was thinking. This is what they call the union. This is for the HT-seq. Well, you can also think about the intersection strict. And then this is gene A, but this is not gene A. This is not gene A, so it's no feature. This is gene A, this is gene A, this is gene A, because it's perfect here, but not here. And this is ambiguous. Or another one. What I'm saying is HT-seq is kind of a quite standard tool. There are others. I mean, and also there are things that you can run directly in R for doing that. But and you can see this figure in this link perfectly. But what I'm saying is there is no one single way of counting things, and it will affect a lot in the follow pipeline. What I do is I use two of them, usually this one, and they correlate quite well. So if they correlate quite well, and I mean they're not that different, that much difference between them, I'm kind of happy. I mean, in general, they don't go too far apart. If they will be completely different if you use this one or this one, then your data is very sensitive to the parameters that you are taking and you're selecting. But if whatever you take, the results are similar, this is not going to be your kind of, it's not going to affect the outcome dramatically. I will put it this way. Okay. This is the HT-seq is the one we use in STATegra. So the count data we are going to use in STATegra is actually HT-seq data. You stay with me. We got the FASTQ files. Then we map to the genome. We filter things that we don't think. Then we say, okay, with those reads, I'm going to assign to genes. And now I come to this situation, right? This situation is, I will improve the quality of the brightness and everything, but those are the genes, and there are many more, and those are the samples, okay? And here what I have, it's the number of reads that goes into this gene for this sample. This is count data. Can I work directly with this data? That's one problem. If we have one sample with 100 million reads and another sample with 10 million reads, I will make different a situation. What else? But one thing is quality control, but even if you have, imagine you say, well, I don't want samples below 10 million reads, fine, but you have one sample with 20 million reads, enough? 20 million reads is a lot. It's kind of, well, a lot not. I would say it's good enough for genes at a certain level, not for isoforms. 100 millions is fantastic. But then the counts cannot be compared because you have more reads here just because you have more reads in your sample, right? Okay, what else can happen? The length of the gene. That's a very good point. If we have a gene like that and a gene like that, just by the length, this one will get more reads and this one will get a lower number of reads. What do you think are the effects of this later on when we do the analysis? Yes, well, one second. You should normalize by the sequencing depth, I agree. So you can say I divide this by my total number of reads or my total million of reads, and then I get a value that is comparable. What saying is different because it's the length of the gene. And my question is, the length of the gene, do you think it will have an effect later on when we do the analysis? And how it affects. So I repeat your. Okay, I rephrase what you're saying. So you have a longer number of a longer gene, you have more reads there likely to happen. A smaller gene, you have, I mean, fewer reads going there. Fine, and then you are less likely to then quantify the smaller one, right? My question is. Yes, you cannot compare between those genes because this is like larger and this is smaller. But later on, when I tell you, when I do the differential expression, which means which is changing between condition A and condition B, will be the length of the gene having an effect? You think so? Who says no? No, okay. Why do you think yes? Okay, I'll let you think. Why do you think not? Mm-hmm. Okay, what if I tell you that, Or what if I ask you that, will the variability of the counting be affected? So the variability will be affected. And if you affect the variability, you affect the ability to identify changes. So the answer is yes. The length of the genes has a bias. And most of the things I'm going to show you now or talk about is about exactly this question. Because with the fluorescence in the macroarrays, with the log transformation, quantile, blah, 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 we will see that quantile is not working here, which was major drawback. The problem when you log transform, and just log transform, is that yes, the highest values get smaller, variability gets smaller, but then the changes in the lower range gets kind of highlighted. And then you have a bias in the other direction. So that's why RNA-seq has been developed a lot in the, and there's a lot of kind of discussion about what is the best way of normalizing differential expression and everything. So keep this in mind. The gen length has an effect. The variability has something to do with that. How we normalize all these questions, this is what I'm going to try to show you now. So count per gene, are they comparable? What is the probabilistic model to consider? Some people were saying like, well, when we have fluorescence, when we log transform them in the macroarray, we were thinking maybe it's a Gaussian distribution, we do the quantile normalization, perfect. But here this is count data, and we should take it as a count data. So let's go a bit into this path of counting data. So if you think about count data, the first thing is there are different distributions for working with the count data. Poisson, negative binomial, many others, okay? Those are discrete distributions. This is one of the first ones kind of comparing the technical reproducibility between RNA-seq and expression, and they say that RNA-seq is more reproducible, etc. And it's John Marioni. Has a huge work, and also now in the single cell normalization. It's really brilliant in many aspects of his work. And they decided here to go for the Poisson distribution. So can anybody tell me what is the Poisson distribution? I don't care about the formulas right now. The Poisson is the number of events that you are expected to happen in a fixed interval of a time of events. So when I was doing the PhD, or on the courses on that, that was kind of very used when you want to think about how many cars will be in a gas station for a given time. Yes, then you have a Poisson distribution, because cars goes and ins in an exponential time, blah, blah, blah. I will not go into that. But this is kind of the idea. How many events occurring in a given space or in a given interval? Or how many events we have? This is the formula of the Poisson. And the only thing with the Poisson is that the mean equals the variance. Higher mean, higher variance. And and they are both they're really, really, really associated. So I will go into that a bit later. So they did an interesting analysis. You remember I was telling you have flow cells. This is a flow cell with 8 lanes, and another flow cell with 8 lanes, and they were comparing things. And they were comparing about if my genes values were really behaving like a Poisson or not, and everything. And they say, well, only for extreme cases this is not entirely true. So they were happy with that. And then what you are doing is for every gene you estimate a Poisson model for the case and for the control, and then you compare the difference between them, right? That's one way of looking into that. Or you can do a generalized linear model. I will go into that, just a model that is not Gaussian, but considers that if the case and control is a variable to consider. And then you can do likelihood ratio test, etc. But this goes into this direction. The problem was that there were some other researchers saying, look, the Poisson seems to be good, but it's maybe not good enough. And the reason is exactly this. The mean is associated to the variance, and the variance is constrained. And what we are saying is that there is more variance that what the Poisson is telling. There is more variance there. So the other type of function able to work with that is called the negative binomial. So I will not ask what is a negative binomial then, but is actually the number of successes in a sequence of independent until you get a number of failures. So imagine I throw a dice. Okay? If I don't get a 1, I say it's a success. So I get throw in, and then I will stop when I get 3 ones. How many successes will likely I have? This is the idea of the negative binomial. And what they tested is that, well, in reality, the Poisson, the negative binomial is like a Poisson in the extreme when, I mean, the probability of error is kind of going to infinity. So they are kind of associated, but the negative binomial has different ways of, I mean, and it's has a larger variance and allows like for larger variance that can be somehow not completely, but a bit more independent than the mean. I'm telling this because this is this will define later how we do the differential expression. We are using negative binomials to think about our population, okay, our count data. If you get this as an intuition, that's fine. There is papers. I put a lot of papers in the Dropbox, and take a look to that if you think. I mean, for me as a statistician, it's like a funny thing to read. Maybe some others are not that excited, but and I mean, researchers like Robinson and I mean Gordon Smith, and they're really good on those parts. So what were the two main kind of packages? There are even more, but I think there's the two ones that kind of set a difference that use a negative binomial. DSEC, now is DSEC2, there is a new version already for some time, and HR. And I would recommend, and this is really a recommendation, I mean, if you have to pick papers from the ones I have been showing, pick this one. Because this one is actually, we are going to run the things that is being said there. And we're going to work with the linear models, etc. Okay? This paper is actually the authors from DSEC and the authors from HR came together and say, well, we have a similar pipeline, maybe different, but this is how things can be done if you work with count data. The difference are here is in the way they compute they normalize the data, in the way they compute what is called pseudo counts. I will come to that later. It's a small of a difference between them, very important, and the outcome of that is DSEC is more conservative, and HR is more kind of it's less conservative. Well, because this is the way they address the variance, how it's computed. I will stop this part here and I will come back in a second. Talking about normalization, differential expression, how we address the data, it's complex because it affects every part of the analysis. So right now, you have to think, if we have count data, the assumption is that the negative binomial is the best way to think about our data. Let's go back one step, and let's go back what you were saying about how we compare genes when we have a different sequencing depth. Okay? So this is exactly the kind of example that you were giving. I just zooming in and saying, well, if I sum all the counts of this sample, if it goes in this one, this one, this one, and this one. And I mean, look, this has much smaller number of reads than this one, right? If this is the case, it's like not 50%, but then those, I mean, I cannot compare this versus this one. And people were saying, okay, how we can make things comparable? And how can we make things comparable if we take the gene length into account? And one of the papers that came out, and this is kind of old by NGS means, Mortazavi, it's one of the PIs of the STATegra project, and is in charge of the DNA-seq data in the project and the single-cell RNA-seq. And Ricardo is his PhD who will be giving the session. But they came out with the RPKM and FPKM. Very intuitive measure. I mean, they wanted to have something that, okay, I want to compare things, how we do that. What is the difference between reads and fragments? We work with fragments, and we can either have one read or have the parent, so 2 reads per fragment. So in this sense, we summarize per reads when you have single end, so only 1 read per fragment, or you summarize per fragments when you have parent situation. But the idea is the same. We want to have the number of reads per kilobase per million. So we have the number of reads, and we divide by the length of the transcript in kilobases, and the million mapped reads. So how many millions do we have? And it's a way to compare. That makes sense for you? Like I have a gene, and I say, first thing, if I have more reads, I want to divide by, 0.17, and here I want to divide by 0.30, or by 30, and here I need to divide by 17, because I want to make them comparable at the million scale or at the 100K scale, but I want to have them all at the same, kind of the same numbers. So basically proportions. The other thing is, the longer the read, the longer the gene, the more reads you're going to have. So I want to divide also by the length of the gene in kilobases. So I consider the total amount of reads per million, so how many millions, it will be 0.27, blah, blah, blah. And I consider the length of the gene, and they came out with this measure. And for this paper, this measure was kind of relevant. They show interesting things. I'm going to show you an example about maybe when things are not that good. The assumptions here, and well, I show you the example and then we discuss about it. I call it problems with RPKM or FPKM. Imagine we have, this is the total mRNA in condition 1 in the cell, right? This is the total amount. And this is my gene X, which has this proportion of the total mRNA. Okay? Then I go to condition 2, and you can see that I have, for all the genes has remained exactly the same, all of them, except gene X. And gene X has grown a lot. So much that it's kind of a decent percentage of the total amount of mRNA. The question is, when I'm doing RNA-seq, I'm picking. So I'm taking 1 read and 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 1 read. When I do FPKM, I divide it by the total amount of reads. But in proportion, one gene here will become lower proportion here. Not because the gene went down, just because this one went up. And that's why FPKM makes things problem. I mean, it's a problem when you have this type of situations. Because imagine this situation. You have the activation of T cells, okay? Then a lot of expression is going. Oh, wait, a lot of expression for all the genes? Or some differential process has, like, part of the genes are going very high, but some others will remain constant. But because we are taking proportions, it looks like they are going down. They are not going down. They are just remain the same, but in proportion they are smaller. That's the problem with the FPKM. So that's the situation. So how we deal with that. Another paper I would like to recommend to read. I mean, all the papers I'm putting here is for recommendations, but I make special emphasis on some of them. The one I said before is really nice. This one is very nice to have this intuition. What is the thing we are measuring here? So imagine this measure, and I will just going to say, I mean, let me explain what I'm doing here. Imagine I have 10 samples, okay? And I say sample 10 is going to be my reference sample. I can do it in another ways, but I'm going to say sample 10 is my reference sample. Then I go for gene in sample 1 and the total number of reads in sample 1, okay? Gene in sample 10 is my reference, divided by the total number of genes in sample 10. And then I do the log 2 and I get the m value. What they say here is like, well, as I was saying, this is a problem. But is this going to be a problem that affects the entire set of genes? Or is it going to be a problem that affects 5% of the genes? If we have the assumption, and this is very important, that most of the genes are going to remain more or less stable, we can take this measure, we can rank the genes based on this measure, and say the ones that are probably moving are the ones on the top and the ones on the bottom, but not the ones in the middle. So I'm going to compare them by computing the average of this mg for all the genes that are kind of in the middle, because those are the ones I'm expecting not to be moving. And this is how they compute a way to compare samples. Imagine if you have only 5% changing and you rank them, this will be in the top or in the bottom, you take them out. Those ones should remain constant. Those ones are the ones that are going to allow you to compute one value that makes kind of comparable between each other. And this is how exactly it works. Compare the weighted average, and then you have the correction factor, which is also called the offset, and I come into that. This offset is for generalized linear models. I'll come to that into a second. It's not the B0 from linear models. It's something different. And you can compute pseudo counts, which especially the author says pseudo counts are for the linear models, but for generalized linear models, not to use like arbitrarily. But one of the main things I think is important under the assumption that most of them are not differentially expressed. What happens in a STATegra dataset? That most of them are differentially expressed, because the cell is kind of shrinking. It's going from acycling to non-cycling. So we should be extra careful when we do such things. So let's take one step back again. And let's say we have the count data, and I was telling you that we can use the logarithm of the counts just to kind of stabilize. It's not working that well. Sometimes it's used for like visualization or things like that. We can use the quantile normalization. That was one of the first things that people tried, but the problem is here is doesn't fit that well the quantile normalization for all the reasons that I have been showing. We can do the RPKM, FPKM, fine. This is still kind of done in many of the places, but for visualization purposes, usually not for differential expression except for some packages. Counts per million. This is another way of normalizing. Instead of the per the length of the gene, you just say how many reads I have per million reads every gene. I can do the log 2 of the counts per million, which is again, and this is quite used for visualization. And then this, the conditional quantile normalization that we will do later in the hands-on, uses this for the normalizing the model. We can do the TMM, and those are the pseudo counts. And this goes for the generalized linear model analysis, considering the error to be negative binomial. Considering the distribution to be negative binomial. And this is for the normalization. Why is so important? Because when we think about what can we do, this type of approaches goes into the linear models, like lemma. Any one of you has run macroarray analysis? The usual thing is you do lemma. Usually you go here and you use something called boom. So if you use lemma, sometimes people has been using that. We will talk later about that. I think I need to start kind of closing a bit. But this is something we are going to try in the hands-on session. The lemma model using a boom correction. Then we have generalized linear models coming either from TMM or from the CQN, and then I will talk about them. Those are the two options. So let's spend 5 minutes maximum talking about what are those two options. I will recommend, and again, this is a strong recommendation. This is already in the, you will get the slides, so you will have a chance to see later, but this paper is also there. Lemma has the user guide for the, and it has huge user's guide, and it's quite good because it also explains linear models, etc. So usually when we have a linear model, we have something like that. We have the variable that we know and the variable that we want to predict, right? And sometimes we say, well, the variable that we know, for instance, is healthy and disease. And the variable that we want to predict is the expression of the gene. Why? Because if the expression is the same between healthy and control, this will not be relevant. But if this is relevant, this will make a difference between healthy and control. Okay? Imagine I have expression of gene 1 depending on like a baseline, plus if you are healthy, if you are diseased, you have this modification. Fine. The important part is this part, the error. The error is those things that we cannot account in the model. There's variability that we cannot explain. We cannot explain the variability, but we assume that this error behaves like a Gaussian distribution. It's normally distributed, it's Gaussian, it's kind of the most classical thing. What is the problem? If we want to use this, we really need to go into, Gaussian transformations. Count data is not prepared for that. So that's why most of the people say, well, it's not clear, we can go there. Let's go into the generalized linear models. So what is a generalized linear model? Generalized linear model, basically, what it says is, I'm going to do something like that, in this case, but I'm going to transform my data so I can use this type of models. I know my data behaves like a negative binomial, but I'm going to log transform my data. This is the transformation I make here, and then I use this, like the linear model, to explain my log transformation of the data. This is what happens in many generalized linear models. You log transform your data, so your original model is actually e to the blah, blah, blah. I don't want to go into full thing. You have a lot of kind of reference for that, and we will do the hands-on on this part. But how many of you has faced any time before like a generalized linear model? 1, 2, 3, 4. Okay. So it's something that is not that common. You need to, I think, put a bit your head on it. My proposal is, you can see that usually there is a time slot in the morning, like 9 to 9:30, that it says that we can have a review or something. I would recommend you try to take a look at it, and maybe Wednesday or Thursday or something, if I get an email saying, really want to go into that, you tell me, and then I take like maybe 15 minutes going a bit more into that part. Because I would recommend first read it, set your mind, then we come here. Okay? It's not too complex, but it has a special. Okay, and then we go to the, when we do the differential expression, we are here, we need to consider which linear models we are doing, like lemma is Gaussian, or if we have a negative distribution, then we go for generalized linear models. That's fine, perfect. This is kind of the assumptions we are going to use. But we need to give the design, right? Because the design will tell us, like I was saying here, expression is a constant plus if you have a disease, is modified based on that. Or if you have the Icarus, is modified based on that. Great. What do you mean by modified based on that? So we need to define this model. To define this model, we need to identify which variables are important. So we need to go back into the design. What variables will you put in the design? I mean, gender, age. In the case of STATegra, we have the time, we have the condition, if it's Icarus or if it's control. But we also have those ones. If you have the experiment today, and then in 2 weeks you do the same experiment and you get another samples, and you process now things 2 weeks, and then in 2 weeks another thing, those are at different times, right? Okay, we are using the same protocol, we are using the same things, but they have an effect. Because it's maybe not the same person in the lab, it's maybe not the same temperature, it's maybe not the same season. It's all those things that, I mean, samples from the same batch seems to have some variability that they share. It was a very important paper coming from the Irizarry lab, showing that for many papers that they were doing macroarray analysis, they were doing the reanalysis saying, I mean, they were looking into the arrays and they identified like, well, most of the things we get is kind of looking, it seems to be associated to the batch. So you include the batch as a covariate and the results change completely. So why is it important then to think about the batch? I didn't put the slide here, but I have a real case example where we had like samples in a first batch, disease and healthy control, all male. Second batch, disease, healthy control, all female. What can you do? You cannot separate what is the batch from what is the gender. That's a big, big problem, because you cannot be able to say anything about disease-specific gender associations, disease and gender kind of having a kind of shared effect or additive effect between them, because you cannot separate what is the batch from what is the gender. And that was a dramatic case. I think we have. That's one of the reasons that whenever we have a collaboration, we want to be in the design right from the beginning. And that would be a recommendation from you. This is things you need to really think carefully. You are investing a lot of time, a lot of money, a lot of resources. If you invest 1 day preparing it, it pays back, I mean, a lot. I mean, there is different things here, like the batch, but then also library preparation. At that time, we can only do like 6 library preparations at the same time. We have 36 samples, we have to make batches of 6, right? We also have 3.5 sequencing runs, so we go to the machine 3 times or 4 times, it depends how you count. And this makes variability. We have the lanes for each one of the cases. So we were including all these things, trying to make as random as possible. I will show you later kind of a plot on that. But we will invest in a lot of time saying, for every one of the library preparations, we want to have a balance design in the times, in control, Icarus, in the 3 batches that we have for the, I mean, 3 times the samples were obtained. And after all this analysis, library preparation was the ones with the highest variability. And because we did this kind of balanced design, we were able to kind of correct the errors, or partially. If not, that was a big mess. And you will see later when we do the analysis of the RNA-seq data. And I mean, that's what I was showing here. And I was saying at the very beginning, put an effort on that. I will recommend another paper. This one is fantastic. They came like a lot of laboratories saying, no, we're going to take really seriously what are the effects. So we are talking the same kind of type of samples, and we are doing X number of laboratories with X number of protocols, with different maybe sequencing length, with different machines, and they put everything, right? And then for every gene, you compute how much variability is explained by your RIM value, by your RNA extraction batch, RNA concentration, blah, 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 cluster kit, sequencing kit, density, lane, residual variation, things that you cannot explain, for every gene. So what we have here is the box plot for all the genes. In general, for all the genes, I mean, of course, what you cannot explain is probably the biology, right? The part that, I mean, or your contrast of interest, etc. But from the things that you can explain that has more to do with the experimental conditions, with the laboratories, with everything, it was very important, the extraction batch. So we have a batch effect. It was very important, the library preparation date. So library preparation was extremely important. And the primer index using, so those things is good to try and be aware of, because those will affect your experiments. This is important, not only RNA-seq. It's also important for DNA methylation. It's important for DNA-seq. It's important for whatever you do. Take this into consideration. And this paper is like a major kind of a study on how things can vary and why. So the output of differential expression. This is if we use the HR, and we will use it later. I think I'm going to stop because those things are kind of, we will do in the hands-on. I mean, what is the meaning of the log-fold change, p-value, FDR? We'll take it out later. Just thinking that it's different if we use lemma, I think we use generalized linear models, and we need to make an interpretation when we look into them. The other thing is I mean, we want to do functional analysis. I mean, either gene set enrichment, Fisher epigeometric network base. At the beginning of RNA-seq, because the differential expression of RNA-seq, at the very beginning, it was clear that it was affected by the gene length. There was methods doing the kind of the gene set analysis, but correcting for the gene length. There are methods like that. I think we are kind of moving a bit far from this, and this is the gene set that we can use. We will have in the hands-on an experience with that. Two last things, and then we finish and we go for lunch. First, this is the paper I was mentioning before. This is the pipeline they are following. I would recommend you take a look and read it. That's really, it's like pipeline. It even has the code. It goes step by step. It has many questions like why we do that, why are we doing this, etc. This is the other one that I think is very important, orchestrating high-throughput genomic analysis with bioconductor. This one is kind of big names. I mean, Martin Morgan is one of the, I mean, is the head of bioconductor, and Wolfgang Haber is one of the big kind of names within Europe about the bioconductor, and many of those people are really involved in the development. Irizarry, Hansen, I mean, Vincent Carey, gentlemen. This gives like a good overview of tools available just to work with bioconductor and the NGS data. It's a fantastic resource. It's already on the Dropbox also, because it goes into kind of more technical things if eventually you want to go into this direction. And saying that, I think we maybe take a break, go for the lunch.